Welcome back, everyone, and welcome to your next unit, which is genetics. I know Mrs. Ford had just covered with you DNA and DNA mutations, and now we're going to start applying that to the world of genetics, and we'll start with simple Mendelian genetics. You've already learned that chromosomes are contained in the nucleus of a cell, and in those chromosomes, you're going to have pairs, and we have 23 pairs that we've been studying. You learned how these chromosomes are separated during meiosis, and you learned how they are being expressed through DNA transcription and translation. And if we dive deeper into one of these chromosomes, we know they're made out of genes, which are sections of DNA that are comprised of your base pairs. So you already have a good idea of this. And so now we're going to start to look at, well, what about these individual chromosomes and what could be different between them when we're comparing different sorts of traits that are expressed in different organisms? We call these words, a lot of times we will say the words genotype and phenotype, and that's a very common way to describe how how a trait is being expressed using these terms. To keep in mind how this works, basically what we have in this case is down here we have our genotype, which is basically where our DNA is, and as we progress further up, and we know we go through transcription and translation, and we get a protein, and then we get the interactions of our proteins, which will provide some sort of pathway, which will then give us an expression of our phenotype. In this case, we have our simple brown mouse here, but we may have some other type of mouse that could be a different pattern, like this mouse here, which is like a blotched pattern, and it is different in this case, and could be even albino or something to that effect. But we know that these things can occur, and basically the key thing here is to understand that the phenotype is what you see, whereas the genotype are the genes that are causing that trait to be expressed. And here are the definitions of those two words. A genotype indicates the alleles that the organism has inherited regarding a particular trait, whereas the phenotype is the actual visible trait of the organism. And so we see here, we have the genotypes of some mice here. We have these letters, this big B, and we have this brown mouse in this case here. Whereas over in our gene, and that's what our phenotype is the brown, but we do see a white mouse down here that has a different genotype. So again, the genotype indicates the alleles that the organism has inherited regarding a particular trait, and the phenotype is the actual visible trait that you see. We also have to understand the word dominant versus recessive when we're talking about genetics. These are common terms, but just as a review, the dominant trait is the trait that's often expressed when the gene is present, whereas the recessive trait is often hidden unless both alleles are present, and we'll get into that term alleles. An example of a dominant trait could be something like tall, and we'll represent that with a large letter or a big T over here, which represents that case of something that is tall. There's our letter T there. Versus a small t, which would represent something that is short. Now, when we're talking about genotypes, and here's a good example of big T, that genotype would then represent a phenotype of being tall. We know that big T is dominant, so if they're together, a big T and a little t, you're still going to get tall as your phenotype. And then finally, if you have two recessives, a little t and a little t, we know that this is the recessive trait, and our phenotype would be short. So on the left side, we have genotypes of these right here. These are all our genotypes for our different possibilities with pea plants, we could say, that are tall or short. Here we have a picture of two homologous chromosomes, and what we often will say is that the particular gene that is being used, it will have two different alleles. And what alleles are the different forms of a particular gene. We have big T, which is tall, and we have little t, which is short. And that's was used from the previous example. But over here, what we have is an example of these two homologous chromosomes. Remember that every single individual is going to have two sets of chromosomes, 1 through 23. And when you're dealing with two sets of chromosomes, you're going to have different alleles for a particular gene. So if a gene, for example, is hair color, you may have a red allele on one chromosome and a brown allele on another. So two different alleles. Hair color is the gene. You may have a brown allele or a red allele. Your height, you might get two alleles for tall. You might have a tall allele from your dad that you inherited and a tall allele from mom. And so we have other particular situations like this lipoma formation, which is lipids in the body and things like that. That could be another trait as well, but you can see there's two different alleles in this person's case. But keep in mind that alleles are different forms of the same gene. We also have what are called homozygous and heterozygous situations. And a homozygous situation is when both alleles are the same. And up here we have the situation where we have these two reddish colored genes that are the same, or the alleles that are the same. In that case, you might have, for example, if we're using, let's say, height, this might be a big T up here, and over here would be a big T also. So those would be homozygous genes where you might have something different for a different allele, and maybe it could be like seed shape. And in this case, maybe it might be wrinkled versus smooth. And so you might have a smooth allele over here, and over here you might have a wrinkled allele, which is different over here. We often use an underscore when the letters are the same for that. So the recessive wrinkled trait would be this one here, whereas the dominant smooth trait might be over here. And that would be a heterozygous situation, and that's when the alleles are different for a particular gene. And again, a homozygous is when the alleles are the same. And so we see the definition over here for heterozygous as well, alleles that are different. An example being big T, little t. Mendel is the founding father of basic genetics, and what Mendel did is he studied a lot of different traits in peas, and so we often use those as our examples. But you can see here, we see that dominant alleles in this case would include tall and round and yellow and green, and then we had pod shape and pod color and things like that. So these are all dominant traits, and then we have our recessive traits down here at the bottom for these particular traits that Mendel studied in pea plants. So what we're going to do is we're going to look at a practice problem here. We'll do this one first, and we have another one that's actually your first problem on your homework assignment, or your assignment, I should say homework, your daily assignment, that is also posted. So let's tackle this first one up here first. And let me give you some strategies for how we're going to use what is called a Punnett square to solve some of these problems. Our first problem is pretty simple. We're going to cross a heterozygous tall pea plant with another heterozygous tall pea plant. So we're going to cross two pea plants that are basically tall, but they are heterozygous. Heterozygous tells me that the genotypes, and sometimes I'll write that right on the problem, we know that it's going to be big T and a little t because it's heterozygous, two different alleles from that definition we learned earlier. And we know that the other plant is also heterozygous, so big T, little t here. So what we can do then is we can use this Punnett square by separating our different alleles and placing them on the side here. So we'll put our one allele here for the one parent. This parent right here is going to go down here. So this parent will come to this side of the Punnett square. Sorry about that. And then we'll have our other allele here. And then over on the top here, we're going to have our other plant is going to come in. So we have our big T here and our little t here. So as you can see, we put those in the perspective spots of our Punnett square. When you're doing your practice problems, you can draw Punnett squares in your notebook or on scrap paper or whatever you need. So you just need a basic box shaped like this, and you put your parents on the outside. The offspring are what are going to be in the inside, and so we see our first cross, we just bring our T's together. And so we see in our first section there, we get big T, big T. In our second one, we're going to get a big T, little t. And down here in this third one, we always put the dominant letter in front if it's part of it. So we have a big T there, and we have a little t there. And then down here, we have, it looks like two little t's. So we have a little t here and a little t there. So now we can look at our genotype ratio, which is what numbers do we get from this particular cross. Okay, so the first thing we do when we do a genotype ratio is we kind of look for our different genotypes. Now, the only genotypes can exist are the pure dominant form, the big T, or you can get the big T, little t, or you can get the pure recessive form, which is the little t, little t. So when we look at the genotype ratios over here of our offspring, we see that we have one big T. We have two, so we got that one checked off. All right, we have two big T, little t's. You can see that one there. And then we have one of the pure recessive, which is that one there. Now, our phenotype ratio is what are these plants going to look like? So what kind of plant do we have here? Well, we know that this is going to be a tall plant. So this is tall. We know that this has a dominant gene or dominant allele. So this one is going to be tall. We have one there that is tall as well. And we know that this one is too recessive. So this plant is going to be short. So in this case, the phenotype ratio for this particular problem is we have 3 tall plants to 1 short. And if we were doing percentages in this case, we would have, if you think about it, 3 out of 4 plants are tall. So that would be 75% tall and 25% short. So those are the two, that's another way to express your phenotypes is in a percentage when you're doing these problems. Let's go on to the next one, and this one is going to actually be on one of your activities for today, your first problem. In mice, and we got our cute little mice up here, some black and brown mice. In mice, black coat color is dominant over brown. So right away, I'm going to put my large big B here, and I know that that is going to be black. And our brown coat color is going to be our recessive because it says it's being dominated by black. So brown is going to be a little b in that case there. A cross between a black male and a brown female, okay, results in many offspring of which 8 are brown and 9 are black. What is the genotype of both parent mice? Use a Punnett square to support your answer. Well, I know that this female here, the only way that I can get a brown female, there's only one way, and that would be to have 2 small alleles or 2 little b's. So we know that at least one of the particular parents has to have a little b and a little b here. So that tells me that. Now, what we're seeing in the offspring is that 8 are brown and 9 are black. That's roughly 50-50 or 2-to-2 in terms of the phenotype ratio. So when we're looking at that, we have to come up with, well, what's going to be the only way that gives us that ratio? If I put down both that the parent that was black was pure black, a big B and a big B, that big B would transfer into all these boxes, and that would be 100% black if we were to run that cross. So 100% black mice would come out of it if the father was pure or homozygous black. Well, we know that's not the case. We know that since we have roughly 8 and 9, we know that it's 50% to 50%. So we know it's a 50-50 ratio. if it's a 50-50 ratio, the father cannot have this large B here, and instead he would have to be a heterozygous or hybrid, another name for that, a big B, little b. And let's see what happens then. If we run that, we see that our big B, little b falls into place here, a big B, little b falls into place here, and then we see our recessives start to show up here, our brown mice, which are going to be down here, little b, and little b, little b. So now we see, in this case, our offspring would have to be, there's our 50-50. So these offspring are going to be black, and these offspring are going to be brown. And now we can tell what the genotype of both parents are. Then we can record that on our Google Doc that you're going to be assigned today. So the parent genotypes for this case are what we're going to take right off of our Punnett square over here. We know that the father in this case is big B, little b, and we know that that father was crossed with the female that is little b, little b. So you can enter that in your first problem and make sure that that is in that green color so we can see that easily. And Ms. Ford and I will take a look at your problems, see how you do. And then tomorrow we will discuss our incomplete and codominance and possibly get into sex-linked traits as well. Hope you have a great day and enjoy your genetics for the day. Thank you.